So CAR T cell therapy, or chimeric antigen receptor T cell therapy, is a novel therapy using engineered T cells to target different tumor antigens. It's FDA approved for acute lymphoblastic leukemia and diffuse large B-cell lymphoma, as well as mantle cell lymphoma. It is also under investigation with very promising results in patients with multiple myeloma. However, this therapy is associated with unique side effects, such as cytokine release syndrome, neurotoxicity, long-term cytopenias. And as we are learning, there is also increased risk of bleeding and thrombosis that we are seeing in patients in our clinics. So this study looks at patients who underwent CAR T cell therapy for lymphoma and acute lymphoblastic leukemia. And we had over 120 patients and evaluated the risk of both bleeding and thrombosis and the risk factors associated with them. So what we found was in the acute phase, in the first 3 months, 9% of patients had grade 2 or higher bleeding events. And most of them were in the first month after CAR T cell therapy. When we looked at thrombotic events, 6% of patients had thrombotic events, and they were more spread out over the first 3 months after CAR T cell therapy. When we looked at the risk factors associated with these bleeding and thrombotic events, the one that stood out the most was a history of neurotoxicity, or ICANS. And this was associated with both bleeding and thrombosis. It was also associated with a low fibrinogen level, increased level of fibrin degradation products, and prolonged prothrombin time. And when we look at our data in terms of, low fibrinogen, prolonged PT or prothrombin time, increased bleeding, this suggests that there is some kind of vascular endothelial dysfunction ongoing. From prior studies, we know that neurotoxicity, one of the mechanisms of that is vascular endothelial dysfunction. So I think what our data shows is that, both neurotoxicity and as a result, bleeding are both mediated by this vascular dysfunction, and both are interrelated. So a history of neurotoxicity should really make us concerned about future risk of bleeding in the next few weeks, and clinicians should stay very vigilant about risk of bleeding in such patients and closely monitor platelet levels, fibrinogen levels, etc. We also found that low platelet levels at the start, the nadir low platelet levels, were associated with increased risk of bleeding. Interestingly, CRS, or cytokine release syndrome, was not associated with increased risk of bleeding or thrombosis. For patients who had thrombosis and received anticoagulation was found to be very safe, and there were no bleeding events as a result of anticoagulation. But again, these patients should be monitored very closely, especially in the first 1 to 3 months after CAR T cell therapy.